me. I'm really excited to get a chance to talk to you today because I'm going to tell you about a program of research that I'm particularly proud of, which is looking at opening clinical trials, making clinical trials accessible to patients in Australia who have a very aggressive childhood brain cancer. And we've just opened a trial this year, so I'm going to tell you all about it, do a bit of a whistle-stop tour through the timeline of getting a clinical trial up and running, because I'm hearing these questions and we get it a lot as well. In fact, working in this area, I get emails from people who are affected by brain tumours, and because they're doing their own research, which I think is wonderful. And they say, oh, I hear you're working in this area. Do you have a clinical trial? When does it start? When can we get on it? And for the last 10 years, I've had to reply, yes, we want to do a clinical trial, but it's not open yet, and I don't know when it will open, which is incredibly frustrating for everyone. You know, you want to be able to give them something, and you can't. But now, since January, I've been able to reply, yes, we have a trial open. This is the doctor you need to talk to. This is the clinical trial coordinator. So it's been an amazing experience. So I want to give you a sense of how that process happened and the big team effort that was involved in this. So we're going to start. So this 10 years is our involvement in the program. But I actually have to start a bit earlier, back in 2001, which is where this idea first sort of started coming about in the research community. And it's all around how we can get your immune system to fight cancer. So scientists sitting in their lab and they're saying, oh, isn't it great our white blood cells can protect us from viruses and bacteria? What do we need to do to get that same effect against cancer? And there's lots of different immune therapies out there, different approaches to this same idea. The one I'm talking about is CAR T cells, which is quite a radical approach because it involves genetically engineering a patient's white blood cells to make them recognise the tumour. So it's a personalised cell therapy, there's gene engineering involved, it's like science fiction. It's a really exciting space to be working in. So here's our white blood cells, and I love this picture, it's like floating through space. It's a cross-section of a vein. So you can see the red blood cells and the white blood cells. We can take these out to doing a blood draw, grow the white blood cells in the lab, do our genetic engineering, and then when they recognise a tumour cell or come across a tumour cell, and this fuzzy grey thing here is a tumour cell in a dish. I'll play my little video. The green is the CAR T cell, and it finds the tumour and it kills it, okay? So it's a real physical process. They're actually punching holes in the surface of the cell membrane. So this is the whole basis of the therapy. Lots of different groups all around the world working on this. It works really well for some B-cell lymphomas, so there's been amazing clinical trials there. And now everyone's trying to get them applied to other cancer types to get the same kind of response. So this is where my boss, Michael Brown, comes in. He's an oncologist at the Royal Adelaide Hospital. He was actually over in Houston, in Texas, doing his PhD. And there they were running a trial in neuroblastoma patients with these CAR T cells. And he said, I want to bring this technology back to Australia because there was very little happening in Australia. There was one other group doing CAR T cells. And in particular, he wanted to work with metastatic melanoma patients, which is a patient group he'd worked really closely with throughout his career and at the time had very few treatment options. So he brought the technology back and we started a clinical trial with those patients. And we showed that it was sort of feasible, it was safe. The patient responses weren't that spectacular. So what we would sometimes see is a disease stabilisation, which means instead of a rapidly growing tumour, the tumour stops for a while, which is encouraging, but obviously we want to make the tumours shrink and ideally disappear altogether. So the next stage was, we still thought there was a lot of promise in the therapy, and we thought maybe there's a better patient group we can be working with. At the same time, a paediatric neuro-oncologist from Sydney Children's Hospital, David Ziegler, reached out to us and he said, I hear you guys have CAR T cells and I really want to use them in my patients. And we said, that sounds great. So we started this research collaboration. We send samples back and forward. And the particular group he wanted to look at is children with DIPG. So this is a very aggressive tumour that grows right in the pons. So this area of your brain controls your heartbeat and your breathing. And for that reason, it is not safe to operate on. So these patients have so few treatment options. They can have radiotherapy, might slow things down for a bit, but that's about it. And that's why he was so motivated to get access to this very new kind of therapy, because he wanted to have something to offer these patients when they came to see him. So we had to do some preclinical testing to show that the scientific basis was sound. And I am showing you a little bit of mouse data here, but these experiments is really critical to prove that the CAR T cells could actually reach the brain and do something about the brain tumour there. So this is a mouse brain hosting a human tumour. So the tumour is shown in red, and you can see in this mouse, it's covered one hemisphere and it's starting to go into the other. And then this is a mouse treated with CAR T cells. So the red is almost all gone. So the tumour cells have been destroyed and the CAR T cells have swarmed in. I always use the word swarm because it's like, you know how bees talk to each other with chemicals? That's what the T cells do. And it's really fascinating and an area we're actively researching. How do they call to each other and bring their friends in? Basically alerting the immune system that there is a tumour here and we need to do something about it. Okay. So that's the bit that I loved, all the preclinical research, and we had the data from the trial we'd done with the melanoma patients to show that we could do it, and we had this preclinical data to show that it made sense to use these cells for brain tumours. And then you hit the paperwork wall, okay? So this is just a summary of the approvals we needed. So TGA is like the FDA, but for Australia, so they approve the use of new therapies and we have to ask them permission to do a clinical trial. They were pretty good. So it did take a long time to put together the data for them, you know, hundreds of pages of evidence that we presented and how we were going to mitigate risks and what our approach would be. And they took about 4 months to review and approve. HREC is your local research ethics committee associated with every hospital to make sure that what you're doing is in the best interests of patients and the community and not unethical. They were pretty good as well. Again, a lot of lead time preparing the documents for them, and then they took about 4 months to review and approve. And then governance, which is, I thought that was it. I thought, you know, we get approval, we get ethical approval, and we get regulatory approval, and we're done. Governance is when the lawyers get involved. So, which I don't mean to be dismissive because it has, if something goes wrong on the trial, it has huge implications for the hospital, right, and the doctors. So that's when you need to check the financial implications, that the hospital can handle the finances and the indemnity. And there's not, I guess, as firm procedures for that. You know, ethics committees meet at a set time throughout the year and you know when you need to talk to them and when you need to get your applications, and that doesn't happen for governance. So I think there was about 4 months of Sydney Children's Hospital and Royal Adelaide Hospital sending emails back and forth about wording in the contract, in the agreement, and so at that point we're pulling our hair out. But in the meantime, we were trying to get ready for the trial, so we can do a lot of prep at our end because we'll be manufacturing the CAR T cells here in South Australia and sending them back to Sydney. So one of the things we had to work out that we hadn't considered before is how we're going to ship them. So we had to find a courier who could safely send the white blood cells collected from the patients in Sydney, at Sydney Children's Hospital, to here. So we do the manufacturing. And then because this is a living drug, so it's live cells, they have to be frozen at a very cold temperature and sent back at minus 197 degrees. And there's pretty much one company that can do this reliably. So you send it back in liquid nitrogen. So we had to test out all those processes. And then when governance approval came through, we were ready, and the trial opened in January of this year. And we had a patient recruited in March, and she had beautiful CAR T cells, and we were all really happy with the process. And then we couldn't treat her because in the 3 weeks it took us to make and test the product, she progressed, which was so upsetting for everyone. Even the couriers were ringing me up and were saying, oh, you know, we were ready to ship and we heard that this happened, and even they were devastated because this is how aggressive the disease is. So she was stable when she was recruited, and then she wasn't anymore, and it wasn't safe to treat her. So what we're hoping, there is one other clinical trial that she can participate in. If that gives her some stabilisation, she could still get the CAR T cells. But when the tumour's growing actively, our treatment is very inflammatory, okay? Those T cells are going to go in and start killing tumour cells, and it's just too risky if she's already having symptoms from her disease. So that was, you know, we all had to go home and just take some deep breaths. But we had a second patient recruited that I just found out about last week, so we're incredibly excited. We're all focused and ready to go, and we will be getting her shipment from Sydney next week. And then hopefully we'll send it back to her and she'll get that treatment in early June. So watch this space. And I just wanted to highlight the role of NRF throughout this project. So they give us grant funding for the preclinical work. so we can do all that lab-based work. But they've also recently given us a grant so that we can treat 6 GBM patients in Adelaide, so adult patients, to complement the Sydney Children's Hospital trial right here at Royal Adelaide Hospital. So that trial is going to open this year as well, and in fact it is open, and we're looking for patients actively. So NRF have been instrumental throughout this project to provide us with that money just to keep us going, because it is quite hard to get federal funding for these sort of very translational, open-ended projects that are phase 1 clinical trials. So thank you very much.
Really good. Any questions for Tessa? Yes.
Just with the GBM trial, is that to do some kind of comparison, or you seriously think that there could be useful for this therapy?
Yeah, we think so. Part of the testing I didn't go into was checking that GBM and DIPG, so these 2 brain tumours, have something on their surface that the CAR T cells can recognise, and they both do. So we think there is a good scientific rationale for working with GBM patients as well, which is why we've opened the adult arm of the trial. Those patients, so with the DIPG patients, we're looking to recruit them as soon as they're diagnosed. With the GBM patients, because there is a standard of care that is more successful, in at least getting patients some benefit, then we'll be looking at recurrent patients, so that who have already had their surgery and their chemotherapy and their radiotherapy, and then at the point of recurrence they could potentially have the CAR T cells. So that's what the Human Ethics Committee have decided is the best approach.
And so for that first patient that unfortunately had progressed, but you'd already made the CAR T cells, so they're sitting in liquid nitrogen.
They are.
So if she does get onto a different trial and does stabilise, is that feasible then to just be able to ship the cells that are already made up to her when she's at that stage where she can receive them?
Yeah, we have. I didn't ship them because the shipping costs $3,000. So I said I'll hold them here, but they are ready to go. And so if she does, and there's a little that we've put in the protocol, there just needs to be a bit of a washout period. So if she was on a kinase inhibitor, which is one of these other trials that is available, I think 2 or 3 weeks she would need to finish on that drug, and then she could have ours, because we just don't want to give people 2 things at once when we don't know how they interact.
One other question from me. With these CAR T-based therapies, you're taking T cells out, the normal T cells out of a patient, and modifying them to put them back into the patient. What evidence is there of how the T cells might change between a healthy individual prior to them getting cancer and a cancer-bearing patient? Presumably the T cells are
sometimes not so healthy, yeah. And this is a really an area we're trying to research in the lab. So your disease can affect how healthy your immune system is, and the prior treatment that you've had as well. So patients often go on steroids, and unfortunately steroids, because they damp inflammation, negatively affect. So it is a bit of balance. I will say that the children, because they're so young, their cells are so healthy. They grow so fast, markedly different from someone who is 60 or 70. With the melanoma patients, they were often older than 60. So yeah, working with children's cells is beautiful.
So that just leaves one more question from me. We've often heard about preserving cord blood, umbilical cord blood, and so there had been a bit of an effort a while ago of any newborn, or some newborn babies preserving cord blood. Would that be some option down the track of being able to then obtain some of that material?
Yeah, definitely. The cord blood banking is really interesting because there's so many things we could potentially do, but because the therapies aren't there yet, the expense of maintaining your child's cord blood cells seems too much at the moment. But I think in the future, yeah.
So you mentioned that the CAR T cells sort of talk to each other. Do they also then mobilise the body's own T cells that aren't modified, and it's a joint response, or is it just the CAR T cells?
So we're really hoping they do. There is some evidence that once you've kick-started an immune response, the other immune cells in your body that you haven't manipulated at all suddenly realise there's a problem, because they do call to each other with these chemical signatures. So that's something we hope to investigate during the trial, whether we actually get something called a bystander response. All the friends come in. So that's something we really want to find out.